Hi everyone. Today I'll be talking to you about putting things the right way round and the identification of upside down jellyfish in Lake Macquarie. So this is part of my PhD thesis, which I've been doing through the University of Sydney and the Australian Museum. And I've also been the recipient of the AMRI Postgraduate Award, which I used for this chapter of my thesis. So jellyfish blooms have been increasing on a global scale, and they're renowned for their bloom and bust nature. So their populations rapidly increase in what's known as a bloom, followed by a population crash known as a bust. It's been believed that jellyfish blooms have been increasing on a global scale due to increased man-made structures, climate change causing increased temperatures, eutrophication, and changes to salinity, and fisheries depleting predators and competitors. Jellyfish have been expanding their range through natural means as well as anthropogenic means, such as shipping traffic, ballast water, or released through the aquarium trade. It's believed that jellyfish are successful in novel environments because they have high reproductive rates and they have a wide tolerance to a broad range of environmental conditions. So my main focus is the upside down jellyfish, which belongs to the genus Cassiopea. They're kind of like the lazy jellyfish of the sea. They spend most of their time upside down, resting on the sediment with their bell, and then their oral arms extended above them. They do this because their oral arms have photosynthetic algae called zooxanthellae in their tissue. And this position exposes that tissue to the sunlight, providing the jellyfish with up to 90% of their nutritional needs, with the other 10% coming from filter feeding. And as you can see from this bottom photo, they are able to swim when so required. So upside down jellyfish have a global distribution. They mainly occur in tropical to subtropical regions, and they're usually found in very sheltered, protected habitats such as coral reefs, seagrass beds, or mangrove forests. They're considered to be globally invasive with a number of expansion records. These records are occurring every couple of years and all over the world, such as Vietnam, Brazil, Hawaii. And they're considered to be successful invaders because they have high reproductive rates and they are able to reproduce both sexually and asexually. So this photo is of one of their invasion events in Brazil. And as you can see, the Medusa stage of their life cycle just blankets that beach. So some studies have even recorded up to 168 individuals per metre squared. So this can have detrimental impacts as the Medusa stage are considered as ecosystem engineers. So what this means is that they can alter community structures. There's a couple of ways that they can do this. Firstly, through direct impacts such as predation, or secondly, through indirect impacts such as competition. Additionally, the jellyfish release their stinging cells into the water column, and this can force fish to leave the area. So upside down jellyfish within Australia. As you can see from these green dots, they're predominantly in the more tropical and northern regions of the country. However, there's this one population in South Australia. That population has been there for over 20 years and is right by one of the power plant outlets. However, my main focus is these three red dots. That's because they're occurring in temperate New South Wales. So the jellyfish were first reported in Lake Illawarra in 2013, followed by Wallace Lake in 2014. Now, Steve and Shane at the Australian Museum completed a study identifying these, which species was occurring here through morphology, and they found one species, Cassiopea nidrosia, was occurring in Lake Illawarra, and another species, Cassiopea maramitans, was occurring in Wallace Lake. Then in 2017, a member of the public reported that the jellyfish were occurring in Lake Macquarie. So this raised the question of which species was occurring and where they may have come from, which created the foundations of my PhD. So worldwide, there are 9 species of Cassiopea that are occurring, at least 4 of which are in Australia. However, Cassiopea is considered to be a cryptic genus. So this means that they're really difficult to distinguish based on morphological characters alone. As a result, I needed to do the genetics to determine where the species may have come from, followed by the morphology to try and identify and put a name to that species. So these were my initial genetic results. Down in South Australia, we found that Cassiopea andromeda was occurring. Up in this green dot at Lizard Island, we had another species. And then we found the Lake Macquarie population was the same as the Wallace Lake population. And then down in Lake Illawarra, we had a separate species. So as I mentioned before, Steve and Shane initially identified the species in Lake Macquarie and Wallace Lake as Cassiopea marimetans, but we wanted to confirm this through genetics. So as a result, we wanted to get specimens from Moreton Bay, as this is where Cassiopea marimetans was initially described and the type locality. So in order to do this, I got the AMF and AMRI Postgraduate Award, and my aim was to acquire specimens from the Gold Coast to use for morphological and genetic comparisons to the population in Lake Macquarie. So I packed up all my field gear in my car, drove up to the Gold Coast. It had to be rearranged a couple of times thanks to the pandemic. But my first visit was actually SeaWorld. This is because they've set up the Sea Jellies Illuminated aquaria exhibit there, and they've got a lab there that they're doing jellyfish research on. So this middle photo is a picture of their Cassiopea culture. So as you can see, it's been really successful. And then this third photo is a picture wandering through their exhibit. It's actually really cool. So as the public wanders through, they can look at the different jellyfish species and then also have a look in at the research that the scientists are doing. So the colleagues there, I was able to talk to them about where I could find Cassiopea, and they suggested these two locations within the Gold Coast. The first being Runaway Bay and the second Coomba Creek. They think that there's two species occurring in these locations. So firstly, I went to Runaway Bay, but unfortunately we weren't able to find any specimens there. And then I went to Coomba Creek and we found 5 specimens there. I then travelled further north to Pelican Waters, as this is the type locality for Cassiopea marimetans, and we were able to collect 3 jellyfish from this location. So this is what Coomba Creek looks like. As you can see, it's a very natural environment apart from this boardwalk. That's me wandering out halfway through to collect a jellyfish. You can kind of get an idea about how shallow that environment is. Once I collected the jellyfish, I took a tissue sample, and then the rest of the jellyfish was put in plastic bags of formalin to fix those morphological characters. So this is what Pelican Waters looks like. As you can see, it's much more man-made and built up. So there was a seawall running around the outside and a lot more private jetties too. The jellyfish were collected in this little cove here. This is a little video of me collecting one of the jellyfish. As you can see, they're only about knee deep. So you just wander through and there's one of the jellyfish pulsing away, which is pretty convenient and cool. So next was the genetics. So we actually had a lot of problems trying to extract DNA from the jellyfish, which we think is because of their mucus. So we collected tissue samples from 2 parts of the jellyfish. Firstly, the tip of their oral arm, and secondly, the gonads. So this is a video of me extracting the gonads from one of the jellyfish. It kind of just slides out from one of the subgenital pits, and then we're able to place it in 95% ethanol. So I compared the genetic sequences through the cytochrome oxidase subunit 1 gene to other populations around Australia. So these are my initial results. So we found that the Pelican Waters population was the same species as Lake Macquarie and Wallace Lake, confirming that this species was Cassiopea marimetans. I then found that Coomba Creek was a separate species, and it was the same species occurring in Lake Illawarra and was closely related to another population in the Northern Territory. So from this, we're able to conclude that 2 species are occurring in the Moreton Bay region and 2 species are moving down the east coast of Australia. So then I wanted to look at how these Australian populations fit worldwide. So I compared them to as many CO1 sequences from GenBank of Cassiopea that I can find, and I created this big complicated looking tree. But there's only 2 sections of this tree that I'd like to focus on. The first being this red box, as this is where the Lake Macquarie specimens came out. So the Lake Macquarie specimens, Wallace Lake specimens, and Pelican Water were all very closely related to sequences from Hawaii, Brazil, and the Florida Keys. Additionally, the South Australian population was very closely related to specimens from Brazil, Mexico, French Polynesia, Egypt, Israel, Hawaii, and Bermuda. This shows how dispersed just one species of Cassiopea can be. So based on the literature, we were then able to assign names, and we concluded that the species occurring in Lake Macquarie was Cassiopea zamacana. So this means that Cassiopea marometans is a junior synonym of Cassiopea zamacana. Additionally, we were able to conclude that the species occurring in South Australia was Cassiopea andromeda. So something to take note of here is how closely related the two species are. So literature has suggested that zamacana is a recent divergence of Andromeda. Morphologically, they look exactly the same. So you kind of need the genetics to be able to identify them, or if genetics isn't available, people have been using their locality. The next section I'd like to focus on is this blue box, as this is where the Northern Territory, Coombabibba Creek and Lake Illawarra specimens came out. They were closely related to specimens from Papua New Guinea, Hawaii and Japan. However, no name has been assigned yet. So for the rest of this talk, I'll be referring to them as Cassiopea sp. So from here, we were lucky enough to be able to use the genetics and confirm that there were two species occurring. So we wanted to see if there were any morphological characters that distinguish these two species. So in order to do this, I examined specimens from Lake Macquarie, Lake Illawarra, Northern Territory, Papua New Guinea, Wallace Lake, and thanks to this grant, Pelican Waters and Coombabibba Creek. So I looked at a number of continuous and categorical variables. So I measured things like the bell diameter, oral disc height, oral arm width, a number of different features. And then all the results were followed with a Permanova, NMDS, and SIMPA to try and see what these differences were and see what characters were driving differences between species. So I'll just talk you through some of the categorical variables I looked at. So firstly, lappet shape. I've shown one of the lappets with this orange arrow. So you can see they're very kind of rounded here. I also looked at the number of lappets per paramere. So a paramere is shown by these orange boxes. Next, I looked at the number of ocelli. So the ocelli is the light sensory organ of a jellyfish. We didn't actually find this was a very useful feature, as this ranged between 16 and 22. I think it's only useful for one species, which is Cassiopea frondosa, where they always have 12 ocelli. Next, we looked at the large vesicles. So the large vesicles, also known as oral appendages, are these large leaf-like structures. So they have two purposes. The first is they provide extra surface area for the zooxanthellae, and the second purpose is they hold casosomes, which are basically pockets containing the nematocysts or the stinging cells of the jellyfish. This means when they're filter feeding and they capture zooplankton, basically these leaf-like structures can fold over the prey and push the food towards their oral mouths. So with these vesicles, we firstly counted the number of them that were occurring in the oral disc, so this central part of the jellyfish, the number of vesicles occurring along the oral arms, and then on top of that, we took the largest vesicle in the center, the largest vesicle on the arm, and we measured the length and width. The final thing we looked at was the branching pattern of the jellyfish. So as you can see, this is one of their oral arms, and then they have lateral branches that come off the side. So we looked at whether these branches were alternating or whether they were pinnate, so in line with each other. Additionally, we looked at the fork at the end of the arm to see if it was equal in strength and mirroring each other, or whether one side was stronger than the other, so stronger in width as well as having additional lateral branches. So firstly, I'll go through the categorical results. So we actually had a significant Permanova showing that there were differences between species, and the Permanova of the different locations within species was not significant. So this was exactly what you wanted. And as we can see in this NMDS, there was clear clustering. So just a reminder, we're focusing on Cassiopea zamacana and Cassiopea sp, which was the Coombabibba Creek and Lake Illawarra specimens. The following SIMPA confirmed that this was driven by the location of the largest vesicle, the oral arm branching pattern, whether the fork in the oral arm was equal in strength, and the lappet shape. So I'll just show you some of these differences. So as you can see here, this is the specimen from Lake Macquarie, and you can see it's got a large appendage right on the fork of the oral arm. Same with the specimen from Pelican Waters. However, this specimen from Coomba Creek, the large appendage is absent, and instead they mainly occurred on the oral disc. The next thing we looked at was the branching pattern. Again, here is Lake Macquarie, and the branching pattern was alternating. This was the same with Pelican Waters. However, Coomba Creek had a combination of pinnate, so equal with each other, and alternating. Additionally, here we looked at the fork. The Lake Macquarie specimen, one side of the fork was much stronger than the other, with additional lateral branches. This was the same for Pelican Waters. However, Coomba Creek, the fork was very even and nearly mirrored each other. Finally, we looked at the lappet shape. The Lake Macquarie specimens, the lappets were very rounded and connected and not very distinct. This was the same with Pelican Waters. Both of them also had this opaque band that was going around the edge. This differed from the Coomba Creek, where the lappets were much more defined with notches in between each lappet, as well as being much less rounded. So next we looked at the continuous variables. So these was things like the length of the oral arms, the number of vesicles. However, before we could go ahead with this analysis, we needed to do a number of regressions, examining how those features were their relationship with the bell diameter of the specimen. When the regression was significant, we then divided that feature by the bell diameter to make sure that feature was significant across the specimens, as we wanted to make sure that any differences we were seeing was due to them being in different species rather than being different sizes. So again, we had a significant permanova. This was driven by the difference between Zamacana and Cassiopea spur. Again, we didn't have a significant permanova between locations within each species, which was really good. The features that were driving these was the number of central vesicles, the number of vesicles on the oral arms, and the length of the large vesicle on oral arms. So just showing you some of these results. So as you can see here, Cassiopea spur has far fewer vesicles on the oral arms compared to Cassiopea zamacana. This is the same for the length of those vesicles. Cassiopea spur was much smaller than zamacana. However, there are exceptions to this rule. And finally, the number of central vesicles was far greater in Cassiopea spur compared to zamacana, which usually only had one vesicle, sorry. So these were this is the summary of the morphological results and the differences we found between the two species. I'd just like to reiterate that this isn't useful on a global scale. These features are only useful for identifying these two species along the east coast of Australia. As I mentioned before, Cassiopea zamacana is very closely related to Cassiopea andromeda. So the features I've talked about here today would be the same in those species. But regardless, this is very useful for identifying the jellyfish without genetics and monitoring their spread down the east coast of Australia, sorry. Okay, so finally, so from this, we're able to conclude that Cassiopea zamacana is occurring in Lake Macquarie and that Cassiopea marometans is a junior synonym of Cassiopea zamacana. We also were able to confirm that two species are occurring in Moreton Bay and moving down the east coast of Australia. We don't know how they're moving down the east coast of Australia. This could be naturally down the EAC or through anthropogenic means, such as the bottom of a polyp attaching to the boat and then a boat going fishing in multiple lakes down the coast. And we also think that there's only one invasion event. This is because the specimens in Lake Macquarie are all very closely related. And as I've been collecting them, I've been looking at the sex of them, and they've all been female. So we think they may be asexually reproducing within the lake. So where to from here? I'd like to combine the results from this chapter with 3 years worth of field data that I've been collecting, as I'd like to have a further understanding of the population dynamics of Cassiopea within temperate environments. I'd also like to have further understanding from this about what's driving their invasion down the east coast of Australia. Finally, it's outside the scope of my PhD, but I'd love to be able to assign a name to the species that's occurring in Coomba Creek and Lake Illawarra, as there's a few names that are floating around but haven't been assigned yet. So thank you for listening to my talk. Again, I'd like to thank the AMF and AMRI Postgraduate Award, as without it, I wouldn't have been able to present the results today. Thank you.